Greetings to you all. Welcome back to our smart video session. This is Mr Karthikeyan, mentor in NEET and all types of competitive classes or biological sciences. Today in this smart video session, I have bought a smart topic for you, and the topic is Hershey and Chase experiment. Hershey and Chase, both members are famous foremost scientists. They have performed an activity on the bacteriophage and bacteria to prove genetic material is transferable from one cell to another cell. So what is that genetic material? In the sense, DNA. DNA is the genetic material. Then this is the genetic material. What is the proof? The proof is proven by the experiment. So this experiment is performed by two scientists. Those are Hershey and Chase. Because this experiment is also known as Hershey and Chase experiment. We knew already DNA and RNA are present in the nucleus. These both are called nucleic acids. But the DNA is more stable than RNA. Why? Because different reasons are there. In one more video, I shall explain to you the difference between DNA and RNA. Here our main content is DNA will be passed or transferred from one cell to another cell. Because DNA is genetic material. So this proof given through this experiment. Let's see. Here these both scientists, Hershey and Chase, they have taken bacteria and also bacteriophage. Bacteriophage in the sense the viruses which they attack bacteria. Those are called bacteriophage. If viruses they attack animals or humans or birds or insects etc., those are known as zoophage. If any virus they will attack plants or plant related things, such type of viruses are called phytophage. Zoophage, phytophage, bacteriophage. For this experiment, these both scientists they have used radioactive phosphorus and radioactive sulfur. Both are radioactive in nature. Let's see.
Is a virus that infects a bacterium. With the help of bacteriophages, Alfred Hershey and Martha Chase in 1950.
These both are scientists, Hershey and Chase. In 1952, they have done this activity.
Hershey and Chase grew some viruses in a medium that can.
First, they have taken viruses and they have grown those viruses, nothing but bacteriophages, in the medium which is contains radioactive phosphorus. And on another hand, same type of one more medium they have taken. That medium contains radioactive sulfur. One medium contains radioactive phosphorus, one more medium contains radioactive sulfur. Let's see.
Contain radioactive phosphorus. Viruses grown on this medium contain radioactive DNA. Similarly, they grow some viruses in medium containing radioactive sulfur. Viruses grown on this medium contain radioactive protein.
This is the main thing. Listen everybody carefully. One medium is radioactive phosphorus. One more medium is radioactive sulfur. In both media, these bacteriophages they were grown. But coming to radioactive phosphorus medium bacteriophage, in its DNA it become radioactive in nature. The DNA which is present, the genome which is present, the genetic material which is present inside the bacteriophage, which is grown in the medium, radioactive phosphorus, this DNA also become radioactive. But in this radioactive DNA bacteriophage, proteins are normal. DNA become radioactive, proteins are normal. Then coming to radioactive sulfur medium virus. In radioactive sulfur medium virus, DNA is normal, but the proteins become radioactive. Are you getting my point? In sulfur medium, in phosphorus medium, DNA become radioactive, proteins are normal. But in sulfur medium, DNA is normal, proteins become radioactive. Never will be confused. Now, if we allow these bacteriophages attack the bacteria, whenever this bacteriophage attack the bacteria, the radioactive DNA is injected into the bacterial cell, but not proteins. Vice versa, if we allow this bacteriophage to attack the bacteria, here DNA is injected as usual, but proteins are not injected or not transferred. In the sense, the final conclusion is always the genetic material DNA is transferable, not proteins. That is the main thing. And finally, DNA is genetic material because it is transferring from one cell to another cell. That is the experiment of Hershey and Chase. Observe.
These two radioactive viruses were allowed to infect bacteria separately.
Here the radioactive DNA is injected, here also injected.
After infection, the viral codes
But
were
this
removed
is
from the
radioactive,
bacteria
this is
by
not
agitation.
radioactive. In both
And the
cells,
viral particles
proteins
were
are not
separated from the
injected.
bacteria by using a centrifuge. Finally, the bacteria were tested for traces of radioactivity.
Here, radioactive traces of DNA.
The bacteria infected with the virus having radioactive phosphorus contained radioactive DNA.
Radioactive phosphorus DNA.
This indicates that DNA was passed to the bacteria from that virus.
But in that bacteria,
The bacteria infected by the virus having radioactive
this is
sulfur
no
consisted of
radioactive
normal
DNA. And also proteins also
sulfur.
not
This
there.
indicates that the proteins were not passed from the viruses to the bacteria.
So according to genetic material, according to DNA, one cell to another cell, the DNA is passed, transferred, not the proteins and remaining contents. This is the main theme of Hershey and Chase experiment. I hope everybody understood. If you have any doubts, please send your doubts in the comment box. Definitely I shall be clarify those doubts. Thank you. Signing off. This is Mr. Karthikeyan, mentor in biological sciences, all types of competitive classes.